The field is moving on, and we are moving with it to more complex data sets that will describe multiple subsets of these responding T cells in infection and vaccination, and what they will require is methods to help visualize these complex data sets. So looking forward, I think that at iCubed we are thinking about what we need is high performance computing hardware to meet the big data needs for data management, transfer, querying, and of course analysis and visualization. We are very interested in internal collaborations with computational scientists. All of us are classically trained as experimental biologists, and the more interaction that we have with people who have the either informatics or scientific computing or programming or computer science backgrounds, which form those three aspects of computation would be beneficial to us. And of course, all of this is important so that we can train the next generation of scientists, and that is something that we are actively doing in the labs, and we would like to do so as well in terms of computing as well. Thank you very much.
Thank you so much, Lenny. That was great. The next speaker is someone who I always refer to as the mother of our cluster hire. I'm just the one who rallied the forces and put the pieces together, but she's the one who wrote the first letter to the administration saying, we need help. So this is Bethany Jenkins. She's an associate professor in the College of the Environment and Life Sciences in the Department of Cell and Molecular Biology.
Thank you so much, Joan. That was a very, very kind introduction. And why at this age do I often feel like a grandmother? Because it's taken us a while to build this community, and it's been very exciting to see this growing capacity, and particularly the addition of new exciting hires like Dr. Zhang, who you've heard from. So I'm going to tell you a little bit about the community that I'm involved with and where we see our data needs going, and how URI can really be a leader in this type of work. So by training, I'm a microbial oceanographer. My community is the broader oceanographic community. And I'm part of a National Science Foundation Research Coordination Network to look at data needs for this community. And so this is a slide from a presentation I gave at a workshop last summer. And as oceanographers, you also saw beautiful data from Dr. Cornillan's lab, but we need to be able to start putting these disparate types of data together. And so I study the genes and the genetic readout in microbes that live in the ocean. So I'm up here in this omics community looking at metagenomes, metatranscriptomes, transcriptomes, genomes, putting those pieces together. But I have to talk to my colleagues that model ocean circulation and think about biogeochemical fluxes. And so these data are going to be needing a way to talk to each other, need a way to come together and make sense to the broader community. So that's one of the future challenges that's sort of a present need now that nobody, I think, has come up with a really good way to address. And so you've heard a lot about this gene sequence revolution. This is one of the newer generation of sequencers. And then, as Kathy and Ing pointed out, this really disparate cost between the cost of generating sequence data and then the computational capacity and cost to handle the data, which is one of our bigger challenges. And then there's just national initiatives that are pushing these types of data. And so this is another project I've been part of that was spearheaded by the Gordon and Betty Moore Foundation, and the sequencing was done at the National Center for Genomics Research. And this is to really get a handle on what protist biology looks like by sequencing the genes that they express. And one reason they chose this approach is protists have bigger genomes than we do. So they're a very, very difficult group of organisms to sequence the full genomes from. And so we're looking at the genetic readout and need ways to start putting these data together. So I'm going to take you on a little journey to highlight the way we use these data to inform our understanding of ocean biology. And so this is sort of a case study from an integrated project in the North Pacific Ocean. I have the students and former students in my lab that participated on this project. And so you can see one of the groups of organisms I study are these beautiful diatoms. They bioprecipitate silica, so they're really interesting to engineers and other folks that are interested in silica nanopatterning. But each of these species is out in the ocean and they're doing different things, and they're an incredibly diverse group of species. So to understand how they function in the ocean, we've got to think about the biology of upwards of 100,000 species that are in the ocean. And these are one of the major contributors to carbon fixation on global levels. So as far as the upper oceans functioning, the functioning of these organisms is extremely important. And so one thing we know, and this is again, this integration of modeling data, this comes from a global biogeochemical model showing parts of the ocean, which a lot of folks think of the ocean as sort of one big fluid surface. Well, it's a mosaic of nutrient regimes. And what you can see here from this predictive model is the areas highlighted in blue are parts of the ocean where ocean biology is highly limited for growth by the availability of iron. So iron is an essential trace element. And so my lab's been really trying to understand how different diatoms from different ocean basins cope with this very limiting nutrient. And so this is data from a recent paper that was a transcriptome analysis looking at what happens when diatoms are experiencing iron limitation. And I'm just showing you, this is how we use this type of data to understand what's going on in the ocean. So this group took all of the genes that were expressed in high iron conditions and low iron condition and compared their relative gene expression patterns. And you can see these very key genes up here that are highly induced by a limitation of iron. And we've seen the same thing in my lab as well. So these then become molecular targets where you can start to follow biological iron status in natural populations, which is pretty exciting. And so this is another way to look at these types of data, very similar thing, very different diatom, and this is data I got from a colleague that's unpublished. And again, these genes popped out in this global data set. So we need ways to compare and annotate these global data so we can find these really important genes that we want to follow. And so my lab's been trying to understand the dynamics of these genes in the environment, and what you can see is that in two different diatoms in these bar graphs, these genes are highly induced. And I'm not going to go into what all of this data means, but one of the things that we used computational biology to help us with is to understand within this big data set from that sequencing project that I was talking about, what the relationship of those genes are amongst all of these different diatoms that have been sequenced. And so this is a sort of family tree, if you will, of one of the genes. And what we were showing for this particular publication was that this protein, the gene encoding this protein, was present across numerous and diverse diatom lineages. Okay, so a lot of us decided to put our tools together, and I was lucky enough to participate in a project in the North Pacific Ocean that was sort of a test bed for starting to put this toolbox together of biogeochemists, trace metal oceanographers, people looking at genomes, proteomes, transcriptomes, metabolomes, all of these things that are coming out, crossing this gradient from the coastal North Pacific out to the open ocean, getting towards very iron-limited waters out in the North Pacific. And so what we did was we used these genes to assay the iron health in these populations across this transect. And what you can see is as we get out here, we've got a lab-calibrated index that is represented by that straight line going across. And what we've got is relative gene expression of a gene that's expressed when iron is very limited. So we know that the biology in these stations, the photosynthetic biology, is starting to show us iron stress. So we went from using a transcriptome to having a diagnostic marker that helps us understand what the natural population is doing. So the next challenge is, can we combine these methods to look at ecological success? And so this is data from a colleague who looks at iron quotas using very, very sophisticated X-ray technology in these different species. And what he was able to show is that he's basically able to follow iron quotas and then using gene sequencing methods, the presence of these different species across these stations. So that was a lot of quick vignette of some of what we do, and without sort of belaboring the sort of science, what are the computing needs behind all of this? And so for me right now, I'm not focusing on the modeling or the biogeochemistry, I'm just focusing on what we need to manage our sequence data. So we talked about storage being an issue. So we need to store the data, then we need to do things with the data. We need to trim it, we need to assemble it, we need to annotate it, we need to compare it to other species, which is pretty computationally intensive. So right now, the back of envelope calculation I did within my group is that